Hey, this is Mr. Horner, and we're going to look at Unit 1, Lesson 2, and this is all about scientific investigations. So the detective story here is they want to know what are some different types of scientific investigations. The two basic types that we know of are experiments and observations. So we've done a lot of experiments in the room, and we'll continue to do those experiments, but you could think about maybe pouring something in a beaker. And then adding something to that solution, so maybe a tablet, and then when it goes in there, maybe it'll bubble. Doing the experiment itself is the experiment, and then watching to see what happens constitutes those observations that we see. The experiments are often based on those observations. So like we said here, the experiment of adding this and kind of watching what happens involves observations of what happens itself. So it's kind of they're all in the same boat, or they're all very closely related. So some different types of scientific investigations. An experiment is really just an organized procedure, so it's very organized step by step to study something under controlled conditions. So we want to keep maybe the temperature the same. Maybe we want to make sure we only add 5 milliliters of the liquid each time as we go through. So we try to control all those things. Many variables are done in the laboratory because these variables are easier to control. So when we do these experiments, we can control all of the variables that we have. Experiments can also be done in the field, but remember, in the field, when we do field work, then that we don't control certain things. So that's kind of the fun part about doing field experiments. It's easier to show how certain processes act in nature sometimes. So if I want to show how a plant grows, it's going to probably be more real world when we grow it out in the dirt than we do if we just bring some dirt in a lab and then put under a grow light, because you have all kinds of other things that happen in the field. The observations, so some scientific investigations involve only just observations, so we just kind of go out and look. And this is just the process of getting information by using our senses. Jane Goodall was a lady who went out and she actually watched a lot of different animals, and she particularly looked at apes, and she really enjoyed doing that. So if we take a model, we can actually make a model, and that model may be part of that scientific investigation. When you built your marshmallow towers, that was a model. So if you remember when you put that together, a lot of you did the triangle thing here, did kind of like a pyramid, and you put that marshmallow on top. This was a model where the top was an antenna, and then this would have been maybe the steel structure that you would have built it, or aluminum, or whatever it was made out of on Mars, if you really thought about it. So that is what a model is. Models are really useful, and they help us study things that are very small or very large or very complex. So this would have been a very large structure if you built it in real life, so we used a model. And then if you think of DNA, remember DNA kind of winds around itself, and DNA is really small. So in order to see it, we actually make models of DNA, and we can draw those pretty easily too, just by kind of drawing that double helix. So those are models. Parts of a whole, so they want to know what are the parts of a scientific investigation. They share a lot of different elements, so here are the basic elements. First one, we've talked about a little bit in the scientific method, and that's a hypothesis. And remember, that is just a guess. So if you have to guess why something works or how something works, that would be a hypothesis. The formal definition is it's a testable explanation that leads to a scientific investigation. So to be useful, hypothesis has to be testable in a practical and in a meaningful way. So we want to make sure that when we test, we're not just testing random things, that we're actually really putting some thought into testing what we're doing. To make sure that our testing is good, we're going to use something called variables, and those are the factors that change. So anytime we have anything change, and that's a factor, we call that a variable. So it can go up or down. So it might be that we change temperature. We can change temperature, and then we can see how many inches a plant grows at different temperatures as we kind of measure that along. So that's changing some variables. The independent variable is called the test variable, and it's the factor that we change on purpose. So we were talking about temperature and plant growth. So what I would do here is the independent variable I might put on the x-axis, and this would be the independent variable, and it was temperature, and we'll call that in degrees Celsius. And then we want to know how high the plant is growing, and instead of inches, let's use our metric system. So let's think about centimeters. So we want to know in centimeters how much growth has there been in the plant. So the independent variable here would be the thing that we're changing, so we're increasing or decreasing the temperature. And then the dependent variable is the outcome variable, so that would be the growth. And it's the factor that changes because we've changed the temperature. If you make the temperature for a plant 0 degrees, so 0 degrees Celsius, it won't grow because that's freezing. If you make it about 27 degrees Celsius, that's room temperature, and that plant might grow a lot. So the dependent variable is the thing that is always called the outcome variable, and it is the thing that changes as we change the independent variable. So let's even draw a graph here. Let's say that my temperature can go from 0 degrees, we're going to go up to 25 degrees, and then let's go up to 100 degrees, which would be boiling. So our plant we know probably wouldn't grow at 0, but it would grow a little bit better all the way up to 25, and then it would probably get too hot for that plant, and then we would boil all the water out of it. So here we've got centimeters of growth, and that centimeters of growth would be the dependent variable, and it depends on what temperature it is as to how much it would grow. So that's the explanation that we can do for independent and dependent variable. Data is the information that we gather whenever we do an observation or experiment, and it can help us in calculating or reasoning why something happens. We can analyze the data and just make sure we understand that relationship between the independent and dependent variable. And based on all of that data, we can draw conclusions about the hypothesis. So if we go back and we think about our plants again, okay, so we said this was temperature and this was growth. We saw that as the temperature rose up to 25 degrees, it grew really well, and then past that, it didn't grow as well. So our hypothesis might have been temperatures grow optimally at 25 degrees Celsius and not so well at 0 and 100. That would be our hypothesis, and if we actually saw it in the graph, we could then make a conclusion to support our hypothesis. When we look at the graph, we are analyzing, and the graph actually contains all of our data. And so that's the difference between all of those different parts. Quality control, there are some characteristics of a good scientific investigation. Certain checks help us to make sure that we have good scientific practices. We want to verify our experiment by repeating it. So I might do that whole temperature of a plant, how much temperature I have. So I'm trying to maintain the temperature for the plant and see how much growth I get. So I might do it in 1 year and I get this graph, and then I might do it again in a different year, and I might get slightly different data where maybe it grew a little bit better at warmer temperatures that year for some reason. So we can verify, and as long as the data looks about the same, we've repeated it and we've replicated what we've done. On the other hand, if the results can also be reviewed by other scientists not involved in an investigation, so they can review it and say, well, maybe we didn't think about how much rainfall there was, or maybe we didn't think about how much sunlight there was for the plants. So there's a couple of things we can do to quality control. One of those is to verify by repeating our experiment and by replicating it, so doing the same thing and doing it over and over. And then we can also review our experiment by other scientists who really don't have any involvement in the investigation. They just want to make sure that you're doing things right. And that is the difference between the two. And that is almost close to the end here. So how is repetition different from replication? We know that repetition is done when the same scientist repeats the study in the same location. Replication is done when other scientists do the study in different locations. So repetition, you can always remember the same word, and then replication, you always have other and different. And so that should help you kind of remember the difference between repetition is the same, scientist, same location, replication is other scientists and different locations is what we tend to think about when we do that. And that is the end of the section.